As you look at this video, I'm going to try to relate long-term potentiation and neuroplasticity to the matter at hand for us, which is how is our brain as part of our body transformed by these changes that we're discussing in the face of this amazing complexity that could not have been understood by evolutionists and are even argued today concerning the idea that this complexity could not have evolved. You see that we're looking at a very small section of a rat's brain, and we have a set of neurons that are functioning together. You see that the blue cells are supportive. The green extension cell is an axon going out from one nerve, and the yellow protrusions are dendrites of another neuron. You see that the synaptic cleft, that space in between the axon and the dendrite, has receptors and proteins on either side, and then an adjacent dendrite also is receiving chemical output from that axon in the synaptic vesicles and in the chemicals in each synaptic vesicle. You see those small circles represent the vesicles, and you see that the chemicals within, approximately 1,500 to 2,000 molecules, are secreted with each one of those vesicles that's allowed to seep into the synaptic cleft. Now, what is happening in this video is through very tiny electrodes, the area between the axon of one neuron and the dendrite of the following neuron are slowly being altered by the expression of the thoughts or the reactions of this mouse. A mouse is being exposed to stimulation. You see the chemical activity inside of each of those nerve extensions, and then the explosion of the chemicals from one nerve to the other. But what you see, and this is shown in this study by the Salk Institute, is that the connections between two neurons, a linked area of long-term potentiation, can become neuroplastically altered in 2 to 20 minutes of exposure and relationship of two parts of two neurons that fire together or that associate. Therefore, the phrase neurons that fire together, wire together, is now able to be seen in live, real-time view, even in the neurons of a small section of a mouse's brain. So now let's look at how this neuroplasticity could affect or could be the result of our habits and our behaviors. This is what is called the pleasure sensing area of the brain, and it is intimately associated with pain, pleasure, anxiety. That's the ventral tegmental area. And you see that the brain structures of thought and sensation and motivation are surrounding that. The nucleus accumbens sits at the front of the VTA, and it's intimately associated with pleasure. You see now and understand what a dendrite is, leading to a neuronal cell body, and then the axon away from that neuronal cell body. And that now you have a stimulus that has been sent down that axon. And in this particular case, what we have affecting the cell membrane of a following neuron is beta endorphin, which is the chemical produced in natural normal pleasure, for example, by a runner's high, or after intercourse, or with spiritual joy that was spoken of in the Bible. We see that when these vesicles connect with the plasma membrane at the end of one neuronal axon to the dendrite of the next nerve, that those now very simplistic, as you can see, vesicles with chemicals release and attach to receptors on the following dendrite at the synaptic cleft. That stimulus now, that runner's high chemical, beta endorphin, which is very much related to morphine, as you might imagine, and very much associated with receptors for drugs like hydrocodone, morphine, and others. Now you see those receptors connect, and you see the effect on the following neuron. Now, what we see here as compared to a beta endorphin is a synthetic opiate drug like heroin, or a derivative of a natural drug like morphine from a poppy. That same opioid receptor, which is there, created by God naturally to receive pleasure, is now hyper and super stimulated and sends a signal through the neuron using these vesicles of dopamine, which are very much associated with pleasure, that attach to the following neuron. There are many more dopamine molecules there, and you have a much greater response with this synthetic artificial drug. But now higher and higher amounts of those chemicals are needed to get the same response from neuron to neuron. In other words, there is a dampening down or an inhibition of the response, just as happens with nicotine or alcohol or any other drug that's either immoral or illicit. Now we see that the patient, when that is not there, has to have higher and higher doses, and withdrawal symptoms occur. A great example of this entire process is nicotine addiction. Those small yellow chemicals are nicotine that's been inhaled through the lung and has now entered the bloodstream. And those blood cells travel with that nicotine and that blood flow, obviously, to the brain. We see that all chemicals, whether they're injected, inhaled, taken by mouth, and absorbed through the stomach, all eventually wind up in the bloodstream. And all addictive chemicals have their actual action within the brain cells. You now understand this synaptic cleft, and now I'm going to introduce you to the idea of a three-dimensional receptor through which almost all, as you saw in the last video with beta endorphins and morphine and opium-related drugs, almost all addiction is mediated through receptor changes. You see these multiple subunit receptors, nicotine receptors, on the synaptic clefts of neurons of different types, connect together, and they form a small tunnel that has a passageway through the cell wall of the neuron. The nicotine lodges, and when enough nicotine molecules lodge, there is a conformational change from a closed receptor channel through very complex proteins that I'll show you a picture of momentarily, that allows an opening and for those nicotine molecules to cause a secretion of an active chemical that sends a transmission to the next neuron, and so on and so on. Now, the nicotine stops at the first nerve. However, the pleasure sensation received ultimately in the central nervous system at the brain level releases dopamine, as almost all pleasurable sensations do. And then we see a transformation of that neuron and the requirement, as you saw before, of more and more of the same chemical to get the same response. The question is, what is that mechanism and how does it actually take place? Well, with nicotine, as with so many other chemicals, when a chemical is introduced artificially into a system that's meant for God-given pleasure or natural pleasure, you see that the receptors increase in number and they decrease in sensitivity, thereby causing the requirement of more molecules and more molecules to cause the same sensation transmission and the same pleasure. And then, sadly, the addiction process and the process of enslavement to that substance has been accomplished and tends to get worse and worse until the cycle is broken through withdrawal and detoxification.